Bismillahirrahmanirrahim. Welcome again, guys. Welcome back to another video lecture by Kami Biology. Today, in this video lecture, we are going to talk about the restriction modification system of the bacterial cell. Okay, and how the bacterial cell can use the restriction modification system. In the previous video lecture, if you guys remember, we talked about the restriction enzyme and we also talked about the properties of the restriction enzyme. Now to start this video lecture, restriction modification, you need to understand the properties and what is actually the restriction enzyme. I already explained it, it is present into my channel. You can go back to my channel and watch this video lecture. Then you will easily get the concept about the restriction modification system. Okay, so let me clean the whiteboard and let me start. So let me write. Okay. Now, what is restriction modification system? Restriction modification system is that system inside a bacterial cell or inside a host cell that when they protect their cell from the foreign DNA. Okay, now what actually happens inside the bacterial cell or host cell, the restriction enzyme is produced and they can cleave the foreign DNA. Okay, but there is also chances that they can also cleave their own DNA. But to protect their own DNA from the cleavage, this restriction enzyme can modify their own DNA by providing the methyl group on it, or to aid or to tag the DNA with a methyl group. Due to this way, the restriction enzyme have no ability to cleave its own DNA, but they have the ability to cleave the foreign DNA. This is about the restriction modification system. Now, let me explain with the help of diagram. What actually happened? Let's say this is the bacterial cell. Okay. And these are the bacterial chromosome, as you can see. Let me write bacterial genetic material or bacterial genome. Okay. Now the bacterial cell is fine and it can be divided and replicated, and the bacterial cell are grow and multiply. Now sometime what actually happen, there will be an invader. There will be invader that attack on the bacterial cell, and that invader is called a phage, or we can say bacteriophage, which is specific to bacteria and they can attack on bacteria. We call that bacteriophage. So let me draw. So let's say this is the phage. Inside the phage there is also a genetic material of the phage as well. Now when they attach on the bacterial cell, then they will inject their own genome. So this red one can represent the genome of the bacteriophage. So let me write phage genome. Now this phage genome have the ability to produce some enzyme or chemical or toxic chemical that can degrade the bacterial genome. Let's suppose in this case they can produce the restriction enzyme. Let's suppose RE that can cleave the bacterial genome, and after that this phage genome will be transcribed or translated or replicated, due to which the viruses copies are made and we get a lot of viruses, and then it will be released out by lytic cycle or by lytic process. Okay, now what actually happen to prevent this phenomena, or to prevent the its own genome, the bacterial genome, then they can produce an enzyme which we call that restriction enzyme. So remember, there will be a competition start between them. They can also produce restriction enzyme and they can also produce restriction enzyme. But as it is the bacterial cell, so remember, bacterial cell will first of all will produce the restriction enzyme and they have the ability to cleave it. Or sometime what actually happen, most of the time bacterial cell can produce the restriction, but some of the time if the bacterial cell is become weak, so this foreign, or if the invader is too much dangerous, means the phage is too much danger, then they have the ability to produce first restriction enzyme and to degrade it. Or not only they can produce a restriction enzyme, they can produce some other toxic chemical or some other material to degrade this bacterial genome. So the competition will be started between them. Okay, so we can also say that, let's suppose if this is the bacterial genome, as you can see. So we talk that they can cleave the, okay. Now remember, these are the restriction enzyme genes. Restriction enzyme genes. So from that genes, it can produce restriction enzymes. So let's say this red one are the foreign DNA. So it will cleave the foreign DNA, as you can see. Degrade. Okay. Now the foreign DNA will be degraded. This is the job of the restriction enzyme. Now this blue are the bacterial genome, this red are the phage genome. Okay. Sometime what actually happen? As we already talked that restriction enzyme need a palindromic sequences. Restriction enzyme need a specific sequences where they can make a cut in a DNA molecule. Let's suppose if the sequences are, G, A, double T and C, double T, double A, G. If these are the palindromic sequences which are found in the bacterial, in the phage genome, so they will cleave it. Okay. If the same sequences are found here at bacterial genome, so they can also cleave their own genome as well, because this may be a chances. Let's suppose at that specific area, this red, if there is the palindromic sequences are found, means PS, palindromic sequences are found at that specific area, then the restriction enzyme will come, bind here and cleave that specific area. There is also, means it may be a chances that they can also cleave its own DNA. Okay, because when, as we better know, the bacterial genome is very huge genome as compared to the phage. So when they produce the restriction enzyme, so instead they can cleave the phage genome, they will directly cleave its own genome. It may be a chances because their genome is too much higher. So the cleavage capability is very, means there is a high chance of the cleavage capability of the restriction enzyme at that specific bacterial genome instead of the phage genome. Okay, although it is also found in the phage genome, but they will cleave its own DNA. It may be the chances. Now, to protect their own genome, because there may be a chances that the bacterial cell will always protect their own genome. They will always protect their own genome. Then what actually happen? This restriction enzyme will be modified. Modified. Restriction enzyme. So restriction enzyme will be modified. And here, many of the restriction enzymes are produced from the bacterial genome or from the bacterial DNA molecule, because here the restriction genes are present. So many of the restriction enzymes are produced. Remember, some of the restriction enzymes have the ability to cleave the DNA, and some of the restriction enzymes have the ability to modify the DNA. Let's suppose we can also, get another restriction enzyme, which is modified version. Okay, so this is a modified version of the restriction enzyme. This will be the restriction enzyme 2, let's suppose. Now this restriction enzyme have the ability, or the main function of this restriction enzyme is to modify the DNA molecule. Modified DNA, or we can say bacterial DNA. Okay. Let's say they can modify the bacterial genome here. How they can modify the bacterial genome? They can use an enzyme what we call a methyl transferase. So they use the methyl transferase and added a methyl group here. Okay, so as you can see, now this DNA, bacterial DNA, is tagged with a methyl group. As you can see, this is the bacterial DNA. Now it is tagged with the methyl group. Remember, when the bacterial genome is tagged with the methyl group, it means that it can give the signal to the restriction enzyme, means restriction enzyme 1. It will give the signal to restriction enzyme 1 that it is the part of the bacterial genome and you don't cleave this one genome. Okay, it is your own part, so you don't cleave this one part. Then it will find the same restriction enzyme, same restriction site here, sequence is here, and then cleave the foreign DNA. Because the foreign DNA is not methylated, that's why there will be a chances that the foreign DNA will be degraded. So this is called restriction modification system. Now sometime what actually happen, there will be, this is only one function, means that we already talked that restriction enzyme have, we already talked, all of you better know, restriction enzyme have two function. They recognize and cleave or modified. Let me once again tell you, the main function of the restriction enzyme is that they recognize and cleave or modified. We already talked it, okay? But it doesn't mean that they will recognize, cleave as well as modify. Now it doesn't mean that they will have two function at a time. They means recognize, cleave, or recognize, modified the DNA molecule. Then there is a question that is usually arises in our mind. Is it possible for the restriction enzyme to perform these three functions? The answer is yes, they can have also the ability. So let's suppose if again we have, this is the restriction enzyme 1 and this is the restriction enzyme 2. So we have if two restriction enzyme, and when they are combined together, as you can see, when they are combined together, then they can make one of the restriction enzyme. Let me draw here, mean a complex restriction enzyme here. Okay, now this is the modified version, or we can say multiple subunit of restriction enzyme. Multiple subunit of restriction enzyme means you have just one restriction enzyme having multiple subunit. So this multiple subunit of the restriction enzyme can have the ability to perform these three functions, but it will not usually happen. But sometimes we can artificially make this enzyme. It is not present inside the bacterial cell, but artificially it is made. This is all about the restriction modification system of the bacterial cell and how the bacterial cell can use it for their protection. So now one thing more that I must tell you about the restriction modification system, or restriction modification system are usually happen due to methylation. So let me write it usually happen with the help of methylation. Okay, now methylation, and there are several enzymes that are available in a bacterial cell due to which the methylation are happen to this DNA molecule. We have different kinds of enzyme like let me write here, DAM, we have SAM, and same DCM. Now these are the enzymes which usually methylate the DNA sequences. Now remember, these enzymes can methylate the DNA, one of the DNA sequences. Let's suppose we have, the sequences like that. Let's say G, T, A, C. Okay, so then DAM will methylate the adenine. So they will put or provide a methyl group to the adenine. Okay, remember, it is not possible directly. They can use the same. So remember, we have both enzymes, same as well as DAM. So let me write DAM with the help of same, they can add the methyl group. Okay, now the DAM can add the methyl group, but the same can, transfer it. This is very important. Remember, this is the DNA sequences, and whenever the enzyme can recognize that let's suppose restriction enzyme can modify that specific site, then they will call upon these enzyme, methylase enzyme. These are actually the methylase enzyme. Then DAM will, come, and the same will carry the DAM. So the same will just scan the DNA with the DAM, and then when they scan the DNA, and whenever they reach to that specific site where the restriction enzyme can bind, after that the DAM will put the methyl group. Then we have another methylase enzyme, we call that DCM. DCM can usually methylate the sequences we have GGT. So in this case, DCM can usually methylate the C area. So here we get just one point, and we just get one idea about that. Methylase enzyme usually methylated one of the sequences, DNA sequences. And once they methylated the DNA sequences, then the restriction enzyme have no ability to cleave the DNA. Okay, I hope you guys get idea about the restriction modification system. But if still you have any kind of question in your mind, you can write it into a comment. Inshallah, I will give you answer as soon as possible. If you like this video, make sure to hit the like button, share this video, and subscribe the channel. And stay tuned, because in the next video lecture, we are going to talk about the types of the restriction enzyme in a detail. Thank you so much for watching.